Our final speaker is Madeleine from Archaeology. She's speaking on testing methods for juvenile sex estimation using long bone diaphysis dimensions.
Awesome. Okay, so sex estimation is a key component in any forensic investigation involving unknown human remains. It's also very useful in archaeological contexts as it provides a more representational view of the population distribution in the past. Juvenile sex estimation, however, is very different from adult sex estimation. The reason for this is because boys and girls grow at a very similar growth trajectory. So here in these two graphs, we have graphs for the growth of the humerus length in both boys and girls, and you can see that they're both very similar. As such, it's very difficult to actually be using methods for juvenile sex estimation, just because they're so similar throughout growth. So there have been a lot of methods that have been developed that have attempted to determine the sex of juveniles, most of them involving the cranium, the pelvis, and the dentition, as these tend to be very sexually dimorphic features in adults. When these methods were tested outside of the population from which they were developed on, they tended to yield very poor results. So in our analysis, we are using our data and testing it against a method developed by Stahl et al. In 2017, which uses the lengths and widths of long bones in order to discern sex in juvenile individuals. So here is just a distribution of our population. All of our individuals included were collected from a collection of individuals of known sex and age from the Lisbon National History Museum. All of our individuals range from birth to about just underneath 13, which is about when puberty starts in kids. Not all of the individuals had all of the features that we were looking for, so some of the individuals were not included in certain methods. So our results actually showed that the methods did not actually correctly allocate the majority of our individuals. So some of the methods resulted in very low allocations, and the average allocation of correctly identified individuals ranged between 56.25 and 70.59% of individuals correctly assigned. This is very poor because that means that the methods are misclassifying between 30 and 50% of the individuals that were tested in our sample. Furthermore, some of the methods actually correctly assigned more males than females, or vice versa. So in one of our actual methods, it only identified correctly 12.39% of females that were tested, but it also correctly identified 90% of the males from that same method. So you can see that there's just a discrepancy there. So that shows that the method developed by Stahl et al., which was developed on a South African population, was very specific to that population. So when we tested it on our population of individuals from Lisbon, it actually ended up not being as accurate as she had originally stated. Because in most forensic cases and most archaeological cases, we're dealing with individuals where the ancestry is unknown, the fact that this method can't be moved between populations means it's not applicable in the field. This also shows that there is very little differences between boys and girls under the age of 13, which makes it very difficult to discern sex, particularly when using the long bones. While there are more sexually dimorphic features in the juvenile skeleton, methods that are correctly identifying sex are still being researched. I'd like to thank Laura Spake for helping run these data sets through R, as well as Dr. Hugo Cardoso for supervising this project.